All right, I'm going to start. All right, so there we go, ATP. Making ATP from your food. So when we talk about eating food, there's like 3 different types of food you eat: carbohydrates, lipids, and proteins. I'm just going to approach it from the carbohydrate perspective because that's the most complicated part of it. So if I explain carbs, lipids and proteins is easier. So carbs, we're really talking about glucose. It's glucose that enters your blood, so like blood sugar, right? So you've got blood sugar, and then what do you do once the blood sugar is in your blood? Right, so the cell's going to take it in, and ideally we want to convert this over to ATP. So there's things that go into this, and there's things that come out. So what goes in is glucose, and then you need oxygen. So glucose and oxygen, and then you're going to make ATP. Go ahead and mute yourself. So glucose and oxygen go in. What comes out? Carbon dioxide and water. So glucose, you've got sugar going in. You also need oxygen. Then you're going to take that and make ATP. And then the byproduct, the product that's not needed, is carbon dioxide and water. And so you'd say, well, we need water. Yes, but I mean, that's the byproduct of the process. So I'm going to write it a different way now. Glucose, as an aside, you should probably know, you don't need to know many of these chemical formulas. Like water, you know, is H2O, and CO2, you know, is carbon dioxide. Glucose is C6H12O6. So remember, the carbohydrates are written in ratios of 1 to 2 to 1. So instead of writing C6H12O6, I just wrote CH2O, just to make it easier what I'm going to do. So it's CH2O, that's the glucose going in, and then also you need oxygen. So there you go, we need going into the process glucose, which I simplified in here. I just put C1H2O1, and then H2O, right? And then what comes out of it is CO2 plus H2O. Now, if you look at this, it's balanced. So like there's the H2O right here between my hands. Oh, you can't see it. So there's the H2O. And there's the H2O. And then there's CO2. Just kind of balance. And when we talk about photosynthesis, it's the opposite. You know, the plant is taking these two things in, and oxygen is the byproduct, and it's making sugar. So, you know, this is respiration, right? Glucose plus oxygen come up with CO2 and water. So that also kind of tells you, it gives you an answer to a question: why exactly are we breathing? Think about what you're breathing in oxygen, O2, and you're breathing out CO2. So the only difference between oxygen and carbon dioxide, if you were just to look up here, is the C, the carbon. It's like oxygen with carbon attached to it. Where did we get the carbon from? Got it from here. So we're eating food, we're taking this food and we're turning it into ATP, we need to get rid of the carbon. So that's why you're breathing in. The oxygen's coming in, we're attaching the carbon to it, and we're breathing it back out. So, all right, I'm gonna erase this. So cellular respiration, there's three steps to it. First one is called glycolysis, glycolysis. So already if you see glyco, you're thinking sugar. Lysis, that we've used that word a couple of times now, it means to break, right? So we're breaking sugar. This is going to happen in the cytosol of the cell. The cytosol is the liquid part of the cell. So, you have like the cell here, there's the nucleus in the middle, there's a bunch of organelles floating around. They're floating around in cytosol. So by cytosol, I mean somewhere in this fluid. Right, and then there's going to be there's other organelles that we are particularly concerned with called mitochondria. That's where the other two phases are going to happen. So the first one, glycolysis, that just happens out here. The other two, Krebs cycle, electron transport chain, they're going to happen in the mitochondria. I'm going to write that down. I was just telling you that the cytosol is like that liquid that all the organelles are floating in.
That's like the egg white of an egg?
No, that's albumin, egg white. The Krebs cycle, which your book might put it down as the citric acid cycle. So it's called Krebs, or I always go with whatever's the shorter to write. But they also call it citric acid cycle. That's the second part of this we're going to talk about. It happens in the mitochondria. And then the last part, so in your book is taking this last part and it's breaking it down into some other steps. I'm calling it the electron transport chain, but it also might read oxidative phosphorylation or chemosmosis. We're going to just call this the electron transport chain. Which also happens in the mitochondria. If you look at the other video I made on this, or if you look even worse, if you look in the book, you're going to see a whole lot of steps. For example, glycolysis is this huge process with different types of molecules being formed, and there's like enzymes that convert each molecule. And I'm not trying to have you memorize all that. It's more important that you understand what's going on. Right? So really what we're doing is we're going to take sugar and we're going to break it down all the way to nothing, to just electrons. So we're going to talk about each of these steps. So glycolysis happens in cytosol, Krebs cycle happens in mitochondria, electron transport chain also happens in the mitochondria. And if you remember the mitochondria, that's where ATP is made, or it's known as the powerhouse of the cell, ATP synthesis, however you want to say it. But that's where energy is made in the cell. So let me erase this and let me start with glycolysis. So glycolysis is happening in the cytosol. We're starting off with glucose. And when we talk about molecules, a good way to measure the molecule is by the number of carbons that it has. It's very common for us to say, oh, this carbon has this molecule has 6 carbons, but this is a 5-carbon molecule or a 3-carbon molecule. Right, so in the case of glucose, it has six carbons. Remember, it's C6H12O6. So it has 6 carbons. So what I'm going to do is just draw circles as like a, well, I'll put C's, I guess, as carbons. So it's a 6-carbon molecule, that's what's going in. Ultimately, at the end of glycolysis, we want to break this up into 2 3-carbon molecules. So we're essentially breaking it in half. So I guess my first question is, when you break something up, what kind of reaction is that?
Catabolic.
It's catabolic. So it's a catabolic reaction. We're going to release a little bit of energy. Not a whole lot. Really, what this step is about it's about getting this glucose smaller. Because glucose is, it's small enough to get into our blood, and it's small enough to get into our cell, but it's too big. This 6-carbon molecule is too big to get into the mitochondria. We can't get it in. So we have to prepare it and make it smaller. So you have carbons, of course, you have hydrogens and oxygens around this as well. Same thing here. So you imagine like hydrogens and oxygens around it. So what your body's going to do is like rearrange the hydrogens and oxygens. From here to here, it's going to rearrange the hydrogens and oxygens. We don't care how it's rearranging it. And it's splitting it in half. And so we end up with these 2 molecules of pyruvate. Which I see them off the board now.
So there's 2 3-carbon molecules.
Yeah, so
of
you just,
pyruvates.
what's that?
There's both of the, oh yeah, okay.
Yeah, so you go from 1 glucose to 2 pyruvate. That's at the end. The book's going to give you a long process. Fructose biphosphate, and it's like converted again and again, and it becomes this molecule called glyceraldehyde 3-phosphate, and then that gets converted to pyruvate. We're skipping all the way to the end. If you take organic chemistry, then yeah, you might have, or I should say like biochemistry, you're going to have some of this. But for now, glycolysis, we're just taking 1 pyruvate and we're splitting it, 1 glucose, and we're splitting it into 2 pyruvates. Each of them are half as big, 3-carbon molecules. Now, when we do that, when we split this into this, we do get some ATP. Not much. We get 2 ATP from this. Let's say all that crap going on here. So we get some ATP, but this is not the big ATP payoff. So we're just preparing this molecule. So at the end of glycolysis, which happens in the cytosol, we end up with 2 ATP and 2 molecules of pyruvate, which are essentially like, you can view them as half of a piece of glucose. They sell this stuff, like at GNC, you can buy it.
What, cytosol?
Pyruvate.
The pyruvate, so.
I don't know why you would do it, but you don't need it, your body makes it. So that's step 1, glycolysis. Before we go into step 2, which is the Krebs cycle or citric acid cycle, there's a little intermediate step. This pyruvate is 3 carbons long. It's still too big to get into the Krebs cycle. The Krebs cycle only wants things that are 2 carbons long. So we have to take a carbon off of this. So we have to convert this molecule here. And so what happens is that we take one of the carbons off, and one of those carbons, so this carbon gets taken off, and there it is, it's gone. It's going to be CO2. So the carbon comes off, oxygen attaches to it, and this carbon is going to start its way out of your body. I always go like this, I don't know why. Like, this is not where all the carbon is, it's everywhere. And I'm not like throwing up something, I don't know why I do that. But you get the point. This carbon's going off. We're going to breathe it out. Now we're left with these 2 carbons, and instead, there's an enzyme that we're going to attach here called coenzyme A. I'm just putting that, COA, that stands for coenzyme A in it. It's just something that's kind of like an enzyme. It's one of your B vitamins. If you ever study like nutrition and you go over all the B vitamins, they all help here. For example, I think this is pantothenic acid. I mean, there's different different vitamins are involved here. So, coenzyme A. So this new molecule, it was pyruvate, and now we call this acetyl CoA. I know it's kind of messed up, but I mean, so it's not pyruvate that's going to go into phase 2. It's acetyl CoA. So we just tweak pyruvate a little bit, make it a little bit smaller, and we stick this coenzyme on it. So we call it acetyl CoA. Now it's small enough to get into the mitochondria. So before we get fully into the Krebs cycle, I forgot to tell you that you're going to see these molecules in your book, and they all start with either NAD or FAD. And you'll see different forms of them. So it'll be called, for example, NADP, NADPH. They'll have pluses and minuses, and that's all about these molecules getting oxidized or reduced. And we never really talked about oxidation and reduction, so I skip it. The important thing that I want you to know is that NAD and FAD are electron carriers. That's it. So when you see them, in whatever form you see them, that's what they're doing. They're grabbing electrons and they're carrying them. Mostly what they're doing for respiration is that they're carrying them from stage 2 to stage 3. They're carrying them from the Krebs cycle. They're grabbing all the electrons from the Krebs cycle, and they're going to carry them over to the electron transport chain. Like you'll see them there. These are vitamins, actually. This is niacin, and this is riboflavin. You don't need to know that, but just to give you an example, like that's why all the B vitamins are involved in this process. But that's a little thing on the side. So, Krebs cycle, that's where we are next. Remember, it's also called citric acid cycle. All right, so it's like you're going to see in the book, it's kind of like a clock going around. And it's got many steps to it. And there's, like this is like citrate and isocitrate, and it's got all these molecules as you go around, and it forms this oxaloacetate. Again, if you sit there and memorize all the words, you lose track of what's going on. What's going on is that there's like, it's like a machine going around, and what's going into it is acetyl-CoA. And I'm putting these well, I'll put 2 carbons here. That molecule is dropping into this. This thing's going around like this, and then you drop an acetyl-CoA into it. And it's going to take it, grind it all up, and it's going to come back around, grab another acetyl-CoA, grind it all up, and it keeps doing that. That's in the mitochondria. So as it goes through, here's 1 carbon that becomes carbon dioxide. Here's the other carbon later on. It becomes carbon dioxide. And then now it's nothing. Then it's going to grab another one. This Krebs cycle is going to grab another acetyl-CoA. Same thing, rip it apart. The important thing, so then what's the point? The important thing that you're getting from the Krebs cycle is electrons. Electrons are a form of energy. We just want the electrons off of this acetyl-CoA. In fact, it's taking the electrons off the hydrogens. So you want electrons. Electrons are energy. It's electricity, right? This is the electricity off of your food. This acetyl-CoA used to be pyruvate. Pyruvate used to be glucose. The glucose used to be your pancakes this morning. So this is the end. This is the very end of your food. It went into your stomach, went into your intestine. Your body broke it up into blood sugar. Then the blood sugar was taken in by the cell, ripped up. Now it's in the mitochondria. That's it. Last stop for the food. Whatever it used to be, it's no longer. It's no longer even a molecule anymore. It's ripped up into atoms. The carbon part of the acetyl-CoA, we get rid of it. We excrete it. We breathe it out. The rest of it, we get rid of it too when we need to. We can like pee it out or however it leaves. But what we really wanted was electrons. And if you think about it, when you break this molecule up, because that's what we're doing, we're breaking this up into nothing. That's catabolic again. So we end up with some ATP. So again, in the Krebs cycle, we get some ATP. 2. That's lanyard. It's just bonus ATP. It's not trying to get ATP from the Krebs cycle. Really trying to harvest electrons. So if you were looking at like another video or a book, you're going to see a lot of, in this cycle that's going around, you're going to see like FAD and NAD molecules. And it might be like NADPH or NAD+, or you're going to see them over here. And that's telling you that this NAD and FAD, they're grabbing the electrons off of this cycle and they're bringing them over to the electron transport chain. So the first one was glycolysis. That's where you get pyruvate. Then we took the pyruvate and we converted it into acetyl-CoA, which was like a smaller molecule. We like tweak the pyruvate so we can get it into the Krebs cycle. Now here it is in the Krebs cycle. Krebs cycle's ripped it apart, and now it doesn't exist. But we got the electrons. So we're essentially pulling the electrons off of your food, pulling the electricity off of your food. We're going to use that electricity to turn on some pumps that are in your mitochondria. So this is like the next step, the electron transport chain is like the grand finale. So let me draw a bigger mitochondria. Thinking about how to draw this. Whatever, I'm not being exact with it, but I'm drawing it like this. All right. So that's my mitochondria. But the point is that there's an inner and an outer membrane. So these lines that I drew, like this membrane around, is just like your cell membrane. And this inner membrane, if I look at these blue lines and I magnify it, it's going to look just like your cell membrane. In other words, it's a phospholipid bilayer. So it's a phospholipid bilayer. It looks just like the membrane of your cells. 2 layers of phospholipids.
Which blue line are you talking about? Just
All of them.
all of them, okay.
Mitochondria is like a weird, they have theories about it. Like we know now, pretty recently that it has its own DNA. So there's DNA, we always know DNA to be something that belongs in the nucleus. But now we know there's mitochondrial DNA, and it lasts longer, and and then there's thoughts that like this was one of the first organelles, and that it was like whatever, maybe a bacteria swallowed another bacteria, and
Evolution with the
they're evolutional theories from like
microscope.
how did we end up with eukaryotic cells 2 billion years ago? And just the answer is probably lying in this particular organelle, the mitochondria. But anyway, what we need to care about is that there's an inner membrane and an outer membrane. And so you have in here, I'm drawing these dots, but these dots are going to represent protons. All right, and so there's protons inside this inner membrane, and there's protons on the outside. So you see how like I'm putting these dots to represent protons, and what I'm trying to show you here is, and I don't know why I keep doing it, what I'm trying to show you is that they're equal. The hydrogen ions, or the protons, they're equal. There's equal amount inside here and outside of here. All right, so that's just normal state. What we want to do, what the mitochondria wants to do, is it wants to take all the hydrogen ions on the inside and it wants to pump them all out. So in this line here, there's pumps. I'll just pretend to mark them. Put in like little black. So these black things represent the pumps. I don't even know if you can see it. Can't even see it on the video, but whatever. There's pumps. There's pumps in this line, right? And so we want to turn these pumps on and pump all the protons out. So I'm gonna not going to write new ones, I'm just going to erase it if we can. We pumped them all out. Now, if you remember, this is active transport. We're going against diffusion. We're putting all of them out here and none of them inside. So that's going against the concentration gradient. So hydrogen ions are being pumped against the concentration gradient. So to do that, we need energy, that's active transport, and we need energy to do that. We happen to have a good source of energy now, protons. All those protons, I'm sorry, not protons, electrons, all those electrons that were just taken from the Krebs cycle, they're going to be used to turn these pumps on, and it's going to pump all the hydrogen ions out. So now they're all out. None of them are inside, all of them are outside. They want to go back in. Right? Diffusion. Diffusion's naturally occurring. So half of these dots that I have out here, half of these protons, they want to go back in. Fine, the mitochondria is going to let it in, but you're only going in through this one gate. That's it. You can all come in, or half of you can come in, but you're coming in here. That's it. That's the only place you're coming in. So when they all come back in, that's like turning a mill. It's like water making a turbine move, or like imagine water flowing through a mill. You see that big wheel start to turn because water's like flowing through it. That's what the protons are like water. Right? So that's powering a machine right here. And the job of that machine, it's not really a machine, I'm just saying it is. The job of that machine is to take ADP and put it together with the P. Because if you take ADP and you put it together with P, that's anabolic. You're building something. You're taking two things and putting them together. So you're making something new, something bigger. So putting these two together, which I'm realizing you can't see, hold on. Putting these two together, ADP and P, that's anabolic. We need energy. We need energy to put those two together. Where are we going to get the energy to put those two together? You might be thinking electrons, but no, it's all of these hydrogen ions, these protons, they're all flowing through, like the water turning the mill. That's the energy to put the ADP and the P together. So right here, hanging out here, you have ADP sitting there, and you have P sitting there. They're just waiting for something to stick them together. So they're all hanging out here, waiting to be put together. When all the protons start rushing through here, that puts them together. And we get actually around 35, so they say about 34 to 38, there's different numbers. You can say 35 if you want, but this is a big ATP payoff. This is where we're making ATP in the electron transport chain. So all of these hydrogen ions are going to flow back in to the inner membrane, and that's what creates it. So this is really, I can trace all this back. So we have ATP, but how did it become ATP? Well, the proton gradient. The fact that we had all these protons on one side and they're flowing through. So I can call that a proton gradient. That's what gave the power to make the ATP. That's H+. Proton gradient gave us the power, the energy to make the ATP. All right, where did we get the energy to even make the proton gradient?
Active fusion.
Electrons.
The Krebs
The
cycle?
electrons turn, yeah. The electrons turn these pumps on. If we didn't turn these pumps on, wherever they are, and shove all the protons on one side, we wouldn't have that gradient. Gradient meaning that, there's a lot on one side and not so much on the other side. So it's the electrons that did that. We wouldn't have had the electrons if we didn't take them off of something.
The pyruvate.
Yeah, pyruvate. In fact, I could have just put pyruvate. And we would never have pyruvate if we didn't have what in our blood?
Glucose.
Glucose. We would have never had glucose if we didn't eat what?
Pancakes.
Pancakes. Probably off screen now. So our pancakes are now the ATP. And even we can follow this further. If we break the ATP up, what are the two parts that ATP turn into when we break it up?
Diphosphate, or ADP and phosphate.
Yeah, so it's ADP plus P. That P has touched my muscles and let my muscles ride on this board. So the energy that allows my muscles to ride here came from the P, which was on the ATP, and you follow it back. We follow it back all the way to the pancakes. And then, really, we could keep going. We're not doing it, but we could keep going all the way back to sunlight. That's the story of photosynthesis. How did we get the pancakes? The wheat. How did the wheat? How did we get all that starch? That's a different story. But this is all the first law of thermodynamics. Energy is not created, energy is not destroyed, it's transferred. This is probably confusing, so I'm going to kind of erase it, but I was just trying to show you that, this is energy being transferred. And the key for organisms is, how do we make that efficient? When I go from pancakes to glucose, when I go from glucose to pyruvate, I'm going to lose some of that energy, but I'm trying not to lose a whole lot of it. I want to keep it, I want to transfer as much of that energy over as I can. So, so we get 34 to 38 ATP out of the electron transport chain, and that's our big ATP payoff. If you see chemiosmosis or oxidative phosphorylation, I'm just throwing everything into this one idea. This is called co-transport. We have active and passive. Active is what you're seeing here. All the dots get put into one side. Then when they start flowing back through here, that's passive. Coming back in now. That's passive transport. So our body did active transport on purpose. We shoved all the protons out on purpose because we wanted them to flow back through. It's like we created a dam of water, and then we start letting the dam go through the turbine to create energy. It's the same thing, that's what your body's doing. It dammed up all of the protons, and then it lets them all flow through one place. It's not really one, but through certain places, and that generates the ATP. It's not really a machine. There's an enzyme here called ATP synthase. That's what you'll see if you look in the book. But I just thought it's better to understand what's going on. So that's it. We've taken our food and we've turned it into ATP. That's glucose. If other molecules like proteins, they can enter at different places. Some amino acids can enter like glucose. Some of them can jump right into the acetyl-CoA. Some proteins, some amino acids jump right into the Krebs cycle. They don't even go through glycolysis. They just enter right into the Krebs cycle. Same thing with fats. We take like 2 carbons at a time, because it's like a long chain of carbons. So you just take 2 at a time, pull it off, and throw them into the Krebs cycle. You could put that right in. You don't even need to go through glycolysis. If you think about the word glycolysis, glyco, sugar. So that's for sugar. Right? Things that we eat that aren't sugar, they can enter this whole cycle a little bit faster. So they're fast tracked. They're going into the Krebs cycle, or they're going, however, acetyl-CoA. Any questions? This is one way to explain it. I have another video which is horrible, but that's another way. And then, of course, you have your book. That's a third way. But what I didn't want you to do is memorize all the other details that I have not talked about. So if I haven't talked about all the different molecules, don't go memorizing them. You're going to waste a lot of energy. You're not going to remember any of it. This, you have a chance. Like, maybe if I see you on the street in 6 months, you might remember some of this. At least the idea, you'll remember the idea. You'll be able to tell me something, right? If I make you memorize all those words, no, I won't know it. You're not going to know it. So I'd rather you learn the idea. Oh, a little late.
Could have got kicked out of it.
Yeah, that's true. That's true. So I have questions on this. One of the students wanted me to give you a, let me stop the recording because, yeah, let me stop the recording.